On behalf of my co-authors, I am pleased to present our research entitled Diagnostic Performance of Two Confocal Endomicroscopy Systems Detecting Barrett's Dysplasia, a pilot study using a novel bioprobe in ex vivo esophageal tissue. This paper is currently published in this issue of the journal Gastrointestinal Endoscopy. The idea for this research came from our interest in developing an accurate and easy to interpret endomicroscopy diagnostic criteria for Barrett's dysplasia. This is a preclinical feasibility study. However, it is also unique in three ways. To begin with, this is the first study to demonstrate the feasibility of using a topical fluorescent glucose analog called 2-NBDG in endoscopic mucosal resection tissue to detect dysplasia. Second, this study presents a new endomicroscopy criteria based on fluorescence intensity from 2-NBDG uptake in dysplastic esophageal mucosa. Third, this is the first study as well to compare the inter-rater agreement and diagnostic performance of the two currently available endomicroscopy systems. That is the probe-based and endoscope-based systems. We have compared these two systems using their respective Miami and Mainz classification together with our newly proposed fluorescence intensity criteria. Based on our previous experiments, we have validated the increased uptake of 2-NBDG in dysplastic esophageal cells. This increased uptake is facilitated by GLUT1 transport mechanism. Based on these findings, we have proposed that 2-NBDG can function as an optical bioprobe similar to FDG in PET scan. Normal mucosa would show absence of intracellular fluorescence. In contrast, dysplastic mucosa would have intense intracellular fluorescence together with disorganized cellular architecture in keeping with the histopathological characteristics of dysplasia. To accomplish our goals in this study, we obtained confocal laser endomicroscopy or CLE images from 16 fresh endoscopic mucosal resection specimens in 2-NBDG. We then systematically obtained images on 4 quadrants in each of the 16 EMR tissues using both probe-based and endoscope-based CLE probes. A total of 64 image mucosal sites were recorded and histologically verified. We then randomly presented to 3 gastroenterologists 64 probe-based CLE images to be classified using the Miami criteria and 64 stacked endoscope-based CLE images to be classified using the Mainz as well as the fluorescence intensity criteria. Inter-observer agreement and accuracy were determined for dysplasia prediction. Our results demonstrate that the inter-rater agreement and overall accuracy in detecting dysplasia was significantly higher for the fluorescence intensity criteria as compared to the Miami or Mainz criteria respectively. The use of an endoscope-based CLE system in conjunction with 2-NBDG in ex vivo EMR tissue demonstrated excellent inter-rater agreement and improved diagnostic accuracy. However, this imaging technique and the proposed fluorescence intensity criteria using EMR tissue will still require in vivo validation. Nevertheless, these findings support the potential of a topical fluorescent analog with CLE to enhance detection of Barrett's dysplasia. To end, I'd like to thank my co-authors, especially my mentor, Dr. Kenneth Wang, for the opportunity to conduct this interesting study. We hope that our findings will advance the application of endomicroscopy and encourage further investigation in the use of 2-NBDG as a novel biomarker for Barrett's associated neoplasia. Thank you.